The World Health Organization has named antimicrobial resistance as one of the 3 most important public health threats of the 21st century. Bacteria can adapt to a wide array of environmental threats, having developed sophisticated mechanisms of drug resistance to avoid killing by antibiotics. As a result, conventional treatments become ineffective. The numerous and varied human, animal, and agricultural misuses and overuses of antimicrobials are key global drivers of these acquired resistances. Bacteria can adapt and acquire new resistance even against novel antibiotics, while resistance to vaccines is rare. That is why vaccination remains a promising intervention to tackle antimicrobial-resistant infections.
At BactiVax, we focus on a variety of bacterial pathogens, which are of great concern in the clinical setting due to their ability to cause disease and multiple antibiotic resistances. These pathogens are able to escape killing by many antimicrobial agents and are major causes of hospital infections worldwide. 6 highly drug-resistant bacteria, referred to as escape, are causing millions of infections and thousands of deaths each year. Tuberculosis is one of the top 10 causes of death worldwide, and burkholderia is a major threat to people with chronic pulmonary disorders, like cystic fibrosis, resulting in reduced life expectancy.
Vaccines have different ingredients that ensure their safety and effectiveness, namely antigens, adjuvants, stabilizers, and preservatives. While they all are necessary, of great importance is the antigen. An antigen is any substance that trains our body to specifically recognize a pathogen and prevent the disease it causes. That is to say, it causes our immune systems to activate white blood cells which will produce specific antibodies against it. Those antibodies will be kept in our bodies so that they can later protect us from the pathogen if we are exposed to it.
The antigen in a vaccine can be either a weakened or kill form of a pathogen, or a small part of it, meaning that it is safe enough to not cause the disease when given to people, but still keeps the ability to trigger an immune response in our bodies. Basically, it imitates the scenario of an infection without causing the disease.
These antigens are carefully selected to facilitate the work of our immune system and ensure long-term immunity. Some of BactiVax projects focus on the identification of the specific antigens in different pathogens and their purification for a future use as a vaccine. For example, by using computer software, we can predict which molecule from the bacteria is the best antigen and has the potential to protect us from the disease. After the first step, we can modify and produce the chosen molecule of interest. Every change that we introduce to our antigen is to increase the capacities of the final product to protect us. We confirm this prediction with experimental work to see if the antigen works as expected.
At BactiVax, we study the immune system and its response to bacteria in order to design next-generation vaccines. The reaction of the immune system towards antigens constitutes the host response, which entails countermeasures against the pathogens containing the antigen. The human immune system contains 2 distinct arms with different objectives. The innate immune system is the first line of defense against infectious pathogens, designed to respond rapidly and non-specifically to prevent or limit infection. This initial response attempts to contain and kill pathogens by engulfing and subsequently destroying them using a multitude of enzymes and other mechanisms.
On the other hand, the adaptive immune system represents a learned response against pathogens. This response develops over the course of the first encounter with the pathogen and endures over months or years, allowing adaptive cells to be triggered again when the body comes into contact with the same pathogen. Adaptive responses are generated by B cells, which produce antibodies against pathogens, and T cells, which mount cellular responses against infected cells. Both cell types are specific to unique antigens, and for this reason, many vaccine strategies aim to produce an adaptive immune response against specific, unique antigens of the target pathogen.
Not all the antigens trigger the same immune response. In fact, some of them do not provoke any response at all, or very few. While some others are immunogenic and do generate antibodies, the protection might not be adequate. Due to this, to get the maximum protection in vaccine development, the right antigen must be chosen.
Understanding the host response and how antigens interact with our immune system and are able to start an immune response is essential for vaccine development. But in order to have a great vaccine, adjuvants are needed. And why is that? Well, these molecules are able to trigger a desired host response for a specific pathogen. Also, they increase the ability of the antigens to induce protection by carrying it out through our blood and releasing it slowly and in a controlled way. That's why they are called adjuvants, because in the end, they help the main component of the vaccine, the antigen, to do its job and provide immune protection against an unwanted pathogen. At BactiVax, we will design and produce new effective adjuvants and study their interaction with the immune system. We hope that this knowledge enables us to optimize the host response to new vaccine antigens.
In order to be administered to everyone, a candidate vaccine has to be tested and validated. This process is long, expensive, and it can be roughly divided in 3 steps. Firstly, antigens and adjuvants that are supposed to become part of the development vaccine are tested on cells. And what does it mean? We take white blood cells from the blood of donors and we make them grow in the lab. Actually, these cells are part of the innate and adaptive immune response mentioned before, and we monitor how they grow and which factors they produce when they are in contact with the new antigens and adjuvants. In this way, we can say at a very early stage of the vaccine development if the proposed antigens and adjuvants are effective and can stimulate an immune response in our body, similar to the one that would happen when the pathogen infects the body naturally. If the results are promising, the vaccine will be further tested in vivo in animals, for example in mice, and finally it will be tested on humans in the so-called phase 1, 2, and 3 clinical trials.
Once we identify potential protective targets and have a better understanding of the host immune response, we must design suitable delivery systems to achieve good vaccine efficacy. This means that we know what message or target we need to deliver and to whom or which immune cells. But we lack the how in order to induce optimal efficacy. Normally, when we think of vaccines, needles come to mind. However, not all vaccines need to be injected. There are other possible routes.
For example, imagine that you send a package. The delivery can be by boat, by plane, by car, among others. Each one will have advantages and disadvantages. For example, the delivery will be faster, safer, cheaper, and you will be able to send more or less cargo. Well, we have to think the same with vaccines, because they can be administered by either oral route, which means by mouth, subcutaneously, which means under the skin, intramuscular, meaning injected into the muscle, intradermal, meaning injected into the skin layers, and intranasal, meaning through the nose.
At BactiVax, we combine cutting-edge technologies to work on adjuvants. Adjuvants not only work to trigger a host immune response, but they can be like a mailman who makes the letter arrive at its destination, and they can guide the antigen to the target cells. They can be a protective packaging, preventing the antigen from suffering any kind of damage when facing the complex path inside the body. Or they can be a tailor, shaping the immune response that is fits best for the specific infection. At BactiVax, we also want to identify mucosal responses. Respiratory pathogens, for example, they colonize the respiratory tract, so administering vaccines to the airway mucosa may offer a better protection against this type of pathogens at the site where they attack.
So using peptide-based technology, we can reproduce the part of the antigen that is recognized by our immune system, which is called epitope. In this way, we can improve the selectivity and the stability of our message, and at the same time, we can narrowly focus the response towards it. To protect our message, it is also possible to deliver it in itsy-bitsy objects called nanoparticles. To imagine how small they are, you can consider that a human hair is approximately 1,000 times wider than them. The surface of this nanoparticle can also be decorated to create a sort of highly specific address where our message should be sent.
In the previous videos, we discussed how to deliver a bacterial antigen to our body so our immune system can learn how to recognize it and fight it. Essentially, we are training and preparing our immune system for a possible future infection. This is called active immunization. However, some people have a very weak immune system, so we cannot always actively immunize them, neither can we give them antibiotics due to the resistance. So we need to think of something else to help these people. You might have heard of antibodies in the fight against COVID-19. Antibodies are like the weapons that our immune system produces and employs to kill a bacteria or a virus. It's like sending reinforcements to a war zone. Giving antibodies to a patient is called passive immunization. Drug-resistant bacteria are a growing challenge, and vaccine development is a very complex process. Through a European collaboration between scientists of different backgrounds, we aim to provide a viable solution to this issue. If you want to know more about the work that we do, please visit us at www.BactiVax.eu or follow us on Twitter, LinkedIn, or Instagram. Thank you for watching these videos.